This is an A-level biology presentation looking at gene expression in cancer. To get started, all you have to do is name the 6 main types of base mutations that you need for the A-level course and describe the impact of each mutation in terms of the genetic code and the polypeptide primary sequence. Pause the presentation, and when you're ready you can restart and move on and check out your answers. The 6 types of mutation can be represented with the 6 pictures in front of you. Originally it was referred to as an insertion mutation, but you should refer to it as an addition mutation. We've got a delete mutation, we've got the inversion mutation, the substitution mutation, we've got a duplication, and of course we've got a translocation. Now in terms of what they will do in terms of primary sequence, is it depends on how many bases are involved in each of the mutations. Remember, if we affect the triplet code, we can have what is called a phase shift, and as a consequence, we can actually knock out several amino acids and change several amino acids with a very small amount of mutation. There are 2 main types of tumors you need to be aware of. Remember, cancer cells are cells that are uncontrollably dividing, and groups of cells that are cancer cells are referred to as tumors. And there are 2 types, benign and malignant. You need to be able to describe the properties and characteristics of a benign tumor and a malignant tumor. Some of the statements in front of you only apply to benign, some apply to malignant, and some apply to both. What you have to do is pause the presentation, work your way through each one, decide if it's benign, malignant, or both, and when you're ready, you can restart the presentation and check your answers. You can, of course, go straight to the answers and then learn out the answers from memory, but if you do learn it from memory, make sure you can associate the statement with benign, malignant, or both. Don't just learn the sequence of the ticks in the quiz. Pause the presentation now, go through your answers whenever you're ready. Pause now. So in terms of characteristics, can grow to a large size is both. Rapid growth is associated with malignant. It has a larger, darker nucleus as malignant because of the large amount of DNA. The cells are unspecialized as malignant. Cells produce adhesive molecules that stick together and stand in the tissues. Actually benign, they're all lumped together in one big lump. No adhesive molecules is malignant because we can have what's called metastasis. We'll come on to that in a second. We've got a no capsule of dense tissue. Well, that's the malignant one. Resultant death is of course both. Most likely to be life-threatening is the malignant one. The effect of cancer is normally localized as benign, treated by surgery. They both can be treated by surgery, but malignant's normally associated with chemo and with radiotherapy as well. Treatment involves both, and high possibility of reoccurrence is of course the malignant. It is the more challenging of the types of cancers you can get in terms of tumors. So we mentioned the word metastasis. And metastasis is the name we give to the process in which secondary cancers are formed. All you have to do in this task is look at the pictures that you're presented with and spend no more than 4 or 5 minutes. Go through and try and describe the process in which a secondary cancer forms via metastasis. After 5 minutes, go through the answers. There's no point in spending more than that time. It's better spent going through and learning off the correct answer rather than trying to work it out. So 5 minutes, pause the presentation, and then go through when you're ready to go through your answers. There are 2 types of genes associated with the formation of cancer. What we start with is what's called a proto-oncogene. And a proto-oncogene is a very useful little gene because that proto-oncogene is able to stimulate mitosis. So whenever cells are needed to replicate, it's proto-oncogenes that turn on that mitosis. However, what can happen is we can have a mutation, and the mutation occurs in one of those proto-oncogenes. And as a consequence, the mutated proto-oncogene becomes what is called an oncogene. And an oncogene works in one of 2 ways. It can either have activation of a receptor protein, so the protein that tells the cell to do mitosis is just constantly supplied, even though there's no growth factor to say you should be doing mitosis, or the oncogene can also produce a large amount of the growth factor, and that growth factor then obviously activates the process of mitosis, and the cell again starts uncontrollable division. Remember, every cell that gets produced is going to be a clone of that original cell, and as a consequence, it's going to be an oncogene as well, and as a consequence, it's going to be cancerous, form cancer tumors. The second type of cancer genes are called tumor suppressing genes. Tumor suppressing genes have a function, as the name suggests, that they go around and they basically stop the mutations developing cancer cells. If, however, something happens to that tumor suppressing gene and it becomes switched off, then we're no longer repairing those mutations in other parts of the cell. As a consequence, it is more likely that one of the normal cells becomes cancerous and forms a tumor. Now, tumor suppressing genes exist in both chromosomes. So tumor suppressing genes to be switched off normally means we have to have two mutations. We have to have a mutation in both of the alleles for that particular gene, in both the maternal and paternal chromosomes. Oncogenes, however, we only need one error mutation in one proto-oncogene to produce an oncogene. So oncogenes are obviously more common because they only need one mutation compared to tumor suppressing genes, and in both cases they are rarely inherited. So in the other presentation on epigenetics, we mentioned the fact that methylation and demethylation can be used to turn on or silence a particular gene. And how that works is, again, the transcription factor, if it's able to bind to a promoter site, will activate transcription of a particular gene. However, if that promoter site is methylated, then the transcription factor cannot bind, and as a consequence, the gene is silenced. Remember, the promoter regions are often referred to as CpG islands because of the repeating CNG pattern. What we can have is what's called hypermethylation. Hypermethylation is just an overabundance of methylation, and if it occurs in particular areas, specifically in the promoter region of a tumor suppressing gene, that tumor suppressing gene is silenced. And if it is silenced, then as we've already discussed, switching off tumor suppressing genes means that we don't have correction of mutations that occur in other parts of the DNA. On another presentation, we do go through the role of estrogen in terms of activating transcription factors that move into the nucleus and as a consequence activate the process of transcription. However, estrogen has also been linked to cancer, and the concentrations of estrogen have been linked quite strongly to breast cancer. Estrogen is able to turn proto-oncogenes, remember those are the genes that are responsible for switching on mitosis, and that estrogen can turn those proto-oncogenes into oncogenes and as a result develop into tumors. Estrogen has been linked to breast cancer, and breast cancer increases after menopause. There's also been evidence to suggest that tumor cells are actually able to increase the production of estrogen, and also the white blood cells that are drawn to the tumors increases estrogen production further, and with the excess of estrogen you're more likely to develop more proto-oncogenes and oncogenes, and as a result more tumor appear. Just to finish off then, thinking about tumor suppressing genes and oncogenes, should people be allowed to test their DNA to see if it makes them high risk for cancer through the NHS? That means they're not directly charged for it, it goes into the NHS budget. Try and come up with reasons why they should be allowed and why they shouldn't be allowed.